So friends, we have already discussed about the causes of somaclonal variations. Now if we summarize those causes into mechanisms of somaclonal variation, then we can summarize them in changes in chromosome number and structure can lead to somaclonal variation. Then mutations in the gene is another mechanism by which somaclonal variations occur. Amplification of DNA or hypomethylation or hypermethylation of DNA will obviously lead to changes in the DNA structure and eventually will lead to somaclonal variation. And last is the activation of transposable elements. We have already discussed that they also lead to the occurrence of somaclonal variation. So these are mechanisms by which somaclonal variations occur. Now how to select the somaclonal variants? Suppose you have regenerated certain plants from the callus tissue or the suspension cultures under tissue culture, and now you want to select in which plants the somaclonal variations have occur. So there are two categories of these methods. First one is the in vivo method and second one is the in vitro method. Now in vivo method, the plants are regenerated through normal regeneration process that occurs during the plant tissue culture, and then they are acclimatized and transferred to the field and are allowed to grow there. And eventually we will screen those plants for their agronomical traits. And when we say agronomical traits, it means either there would be a difference in their pigmentation, there would be an increase in the aerial biomass or increase in the number of the leaves, there might be extensive branching is there, and so on and so forth. So the plants are then screened for the desired trait or the variations that you are expecting to occur in the regenerated plants. Now the major disadvantage of this in vivo method is that this method is very time consuming, because you have to regenerate the plant under in vitro condition, then you have to acclimatize them, and then you have to allow them to grow in fields, and once these plants are grown in field, only then you can screen those plants. Therefore this is highly time consuming process, and it requires screening of many plants to find out the somaclonal variant. So this is just a simple flow chart which exhibits how this in vivo method works. So explant material, shoot regeneration via direct or indirect organogenesis, then this shoot is multiplied on the culture, rooting is there, then hardening of the plant into the field is done, then you go for the screening on the agronomic traits of that particular plant. Now we will discuss about the in vitro method. The first few steps are same. We select a suitable explant material which is grown on the suitable media. Then before we further discuss this method, the point worth highlighting is this is largely done when you want to produce a regenerated plant or a variation that is related to resistance towards specific pathogen or disease or toxicant. So an appropriate explant material is selected and placed onto the suitable media. Now the resistance against a pathogen for which you are performing this tissue culture experiment is selected, and that particular species, from that particular pathogen species, toxins are isolated, and those toxins are then added to the media, and using that media those explant material are grown. So the resistance towards toxicity is tested in vivo, followed by the agronomic values for the first and the second generation. So you can easily then select the variants which were resistant towards that particular toxin, and we can say that the regenerated plant variants which are resistant toward those or which grow properly under the field conditions were resistant toward that particular toxin obtained from a specific species of that pathogen. So again, this is a simple flow chart which explains the entire in vitro method. So explant material, callus is generated, multiplication of callus is there, we select the pathogen, we extract out the toxin from that particular pathogen, these are inoculated on a suitable media, then selection of the tolerant callus is there. So if the callus cells, they develop somaclonal variations in them, and they become resistant toward those toxins, they will proliferate. So we select those tolerant callus cells and we regenerate the plants from those calluses. So this is how in vitro method works. Now we move on to the various methods by which you can detect whether the somaclonal variations have occurred in the plants or not. So the various methods are morphological methods, wherein we explore, we evaluate certain morphological traits of the regenerated plants. Then physicochemical and biochemical markers. There are certain marker. When we say marker, it means those compounds or those enzymes or those protein levels which will have which gives an indication whether that desired variation has occurred in the plant or not. So physicochemical and biochemical markers, by evaluating these markers, we can determine the presence or absence of these somaclonal variations. Then certain cytological methods are there by which you can also determine or detect whether the somaclonal variations have occurred or not. Protein and isozyme markers. So level of the proteins, level of the isozymes, their activity, all will indicate whether the somaclonal variations have occurred in the plant or not. And last is the molecular DNA markers, which give you the most precise or accurate results in terms of whether the variations have occurred in the regenerated plants or not. Now we will discuss each method in detail. So morphological method, as I told you, it depends upon observing the morphological characters of the plant, and this is one of the most widely used methods. And when we talk about the morphological characters, then these include stature of the plant, whether the plant is stooping or whether the plant is erect or whether the plant is bending. So all such kind of characters are observed, along with the height of the plant. Aerial biomass we have discussed. Morphology of the leaf, whether the plant margins are entire, whether the plant leaves they have any different kind of margin. Venation is observed, arrangement of leaf is observed, then pigmentation of the leaf is also observed, which gives you an indication whether there is any kind of somaclonal variation has occurred in the regenerated plant or not. There are certain advantages of this method because you just have to observe the morphological characters, so no laboratory is required. Now this can be done by naked eye. You don't require any kind of magnification equipment, or you don't require any kind of sophisticated equipment to observe, to identify these variations. Then these kind of observations have been found suitable for the preliminary detection. These kind of variation will give you a clear-cut idea whether the variations have been occurring in the regenerated plants or not. There are certain limitations as well. Now these morphological traits are often influenced by the environmental factors and may not reflect the true genetic composition of the plant. Obviously, whenever the plant is exposed to different environmental conditions, the morphology of that plant changes, either in terms of the biomass we talk about, either in terms of the pigmentation of the leaf or the size of the leaf. So all such parameters, they change with the environment in which the plant is grown, and therefore they do not give you a true picture about the genetic composition or the genetic variation in the plant, whether it has occurred or these changes are due to the environmental factors. Then the markers used for phenotypic characters are limited in number and often regulated by the plant development. The detection of various variants using morphological features is often more feasible for perennial plants, because these perennial plants grow over years, and over years you will be better understand the variations. So they largely more suitable for the perennial plants, but in case of other plants, annual plants, such kind of morphological features sometimes are not useful to detect these variations. Now the variations that are observed could also be due to physiological changes induced by the in vitro culture environment. So if you are regenerating the plant under in vitro conditions. So if you are not giving suitable conditions, these variations can occur due to those physiological changes. Further, it has also been found that these variations may not appear immediately in the plants that are regenerated from the tissue culture due to recessive mutations, but they may appear in the subsequent generations that are generated from this regenerated plant. So sometimes this may give you a false negative indication of whether the somaclonal variations have occurred in the plant. So one has to be very careful while dealing with these morphological methods. Second is the physiological and biochemical markers. There are certain advantages over the morphological methods. They are comparatively faster and can be assessed at the early stages of the plant growth in comparison to the morphological parameters, which largely depend upon the growth stage of the plant. Now, if we talk about the examples of physiological markers, these include light or the low temperatures. Now, photoinhibition is a property wherein the photosynthesis gets inhibited when the plant is excessively illuminated. So, if you want to develop a resistance against this photoinhibition, then light becomes a physiological marker. Similarly, the growth of the plant or the processes of the plant are impacted by low temperatures. So, if you are looking for developing a plant species that is resistant towards these low temperature, it becomes a physiological marker. Then biochemical markers. So, analysis of different pigments like chlorophyll is there, carotenoid or anthocyanins, these all constitute the biochemical markers. Similarly, the ability of the plant material to convert carbon dioxide into useful derivatives is nothing but the CO2 assimilating potential. So, this also acts as a biochemical marker. Now, when you are determining, because if you are determining the chlorophyll content and if you are determining whether the variation has occurred or not. So, in some plants the variation may lead to excess of pigmentation, and in some variation there would be a hypopigmentation, and therefore analysis of these pigments become a biochemical marker. The major drawback of such kind of analysis is the complex nature of the method, as well as sophisticated equipments have to be used to get a true picture or the analysis of these pigments. And once you are dealing with sophisticated equipments, high expertise of using those equipment, high expertise in performing that complex method should be there, and that is required. Next is the cytological methods, and in these methods variations can be detected by identifying the chromosomal variations, and these variations can be either in the number of chromosomes or in their morphology. Now, the major advantage of these methods is they will give you a quantifiable data, a data which can be quantified, and the major disadvantages, these methods are time consuming, laborious, tedious, complex or cumbersome. You have to follow a specific method in order to separate the chromosomal fraction without destroying the chromosomes. Then since you are dealing with such kind of method where high expertise of technical skill is required, so a normal person cannot perform such method. You need technical experts having specific skill to perform this methods are required. Another technique that is used for cytological method is flow cytometry, and the use of flow cytometry has revolutionized the usage of these methods. Now, it is considered a very good alternative that measures the changes in DNA content and nuclear characters in the regenerated plants. So, what happens is the DNA or the nucleus is separated from the plant cells, and then they are reacted with a suitable dye which emits fluorescence. Then that mixture is passed through the cytometer, that is why it is known as flow cytometer, and when this mixture passes through a beam of laser light, the fluorescence is detected. So, if we look into the process, that is largely divided into two steps: sample preparation and measurement of the nuclear or the DNA content. So when we talk about sample preparation, it involves the separation of the nuclei or the DNA content. So the plant tissues are chopped in a buffer solution to release the nuclei. Now this nuclei is then bound with a fluorescent dye or a DNA specific dye, and the most commonly used dye is propydium iodide, which is also known as DAPI. Now these fluorescent dyes, they bind with the nuclear DNA, causing their staining. Second step involves the measurement of this nuclear DNA content. Now these stained nuclei, as I told you, they are passed through the flow cytometer through a laser beam, and once the laser beam falls on these dyed nuclear DNA, fluorescence is emitted, and that fluorescence is then detected in the form of a histogram, and the intensity of the fluorescence would be directly proportional to the amount of the nuclear DNA. So these kind of fluorescence, these kind of histograms are obtained whenever such kind of dyed or the stained nuclear DNA are passed through the cytometer and they are impacted by a laser beam, and these histograms are always plotted in the in terms of fluorescence intensity versus the number of nuclei. So higher number of nuclei, higher will be the intensity. Now if you look into this image, this will clearly show the example how cytometry is beneficial in assessing the variations. So a comparison of histograms from the tissue cultured plant and that of the mother plant is done, which will give you a clear cut idea whether there is any kind of change in the chromosome or the number of nucleus in the cell. If you look into the picture A and C, you will see two peaks in G1, two peaks of the proteins in G1 and G2 phase. Similarly, in picture C, G1 and G2 phase. You can clearly see the variation in the intensity of those peaks. Likewise, in the image B and D, you can see the number of the nucleus present in the cells. In the image B, the number of nucleus is higher in comparison to the number of nucleus in the image D. So this is how flow cytometry gives you a clear cut idea about the variations that have occurred in the regenerated plant. So if we talk about the advantages, this method is convenient, involves convenient sample preparation and analysis, and this method is quite efficient for large scale studies. When we are dealing with the studies on ploidy level, even the smallest modification in the chromosome number can be detected by those histograms when it is compared with the mother plant material. So this is how this we can say that flow cytometry is very sensitive in terms of detecting even the smallest variations in the number or the levels of the chromosomes. There are certain limitations as well. It has been found that sometimes the systolic compounds interfere with the quantity or the DNA staining in the cells. So because the evaluation of flow cytometry largely depend upon the staining of the nuclear DNA. So if this process is interfered by some other systolic compounds, that may give you the false results. Now absence of DNA reference standard is another limitations by which you can compare the histograms. So these are few limitations of this method. Another method for detection is the use of protein and isozyme markers. Now these are one of the most widely used markers for identifying the genetic variations. Now depending upon the number of loci, their state and a specific isozyme used, and these variations are determined in terms of the bands that are visualized, and the polymorphism of the bands reveals the variations. Now how these bands are developed? These bands are developed while using gel electrophoresis techniques. So various bands are developed on the gel electrophoretic technique, and you can compare those bands, which will give you a clear cut idea that how they vary. So in this image you can clearly see, I have just shown a representative image of a gel electrogram, phoretic gram. So when we are profiling the protein as the marker, changes in total soluble protein reflect the genetic or epigenetic variations. And these variations, as we discussed, can be detected using SDS page, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis. So you can clearly see there is a variation in the number as well as the intensity of the bands that separate on the gel electrophoresis in the gel electrophoresis method. So any kind of alterations in the banding pattern, whether the new bands emerge or their bands are missing or there is shifting in the position of the bands, that will indicate the variations. Similarly, isozymes, when they are used as the markers, again gel electrophoresis or starch gel electrophoresis is used, and the enzyme activity is visualized using specific staining technique. Again the parameters are same, change in the intensity of the band, number, position or the migration of the pattern also indicate the genetic mutations, or there can be changes in the expressions of the genes, or there can be changes in the chromosomal rearrangement, and all such kind of changes are reflected in terms of the band intensity, number of the bands or their position, or the change in the entire pattern on the electrophoretic method. Now there are certain advantages of using protein markers or isozymes as markers. Now these analysis of proteins or isozymes are useful even in detecting the silent mutations. Now we know that whenever there is a stress under in vitro condition, physiological stress is there on the cells, they induce certain changes in the protein expressions. So by using proteins as marker, you can quantify the variation in the protein expression, or you can quantify the level of the somaclonal variation that occurred due to change in the protein expression. Post translational modification can also be detected. So this is a very cost effective methods which is simple and rapid. So no need of DNA extraction is there in when you are using protein or isozyme as a marker compound for analysis of the somaclonal variation. There are certain limitations of this method as well. Now this method is less sensitive than molecular markers, which we are going to discuss ahead. There are different molecular markers that can also be used to detect whether the somaclonal variations have occurred or not. Environmental and cultural condition. So you are performing an experiment of gel electrophoresis, what kind of temperature or humidity conditions you are exposing to will impact the final results. So these results are dependent upon the conditions of the gel electrophoresis you are using, or the cultural condition which can also influence the protein expression or the isozyme expression. Such kind of method cannot detect the neutral mutations if they are occurring in the regenerated plants. So these were few of the limitations of using protein or isozymes as the markers for detecting somaclonal variations. Now we move on to the detection method where we can use molecular DNA marker. Now there are advantages of using molecular DNA markers for detecting somaclonal variations, and these advantages are DNA based tools used to detect genetic variation, because we are using DNA markers, so we can say this analysis or detection is largely based on the DNA on the DNA based tools. DNA from any tissue can be used for the analysis. So you can use any tissue from the regenerated plant and you can extract out DNA markers from that and you can analyze. Now such kind of DNA markers that are used, they are independent of environmental influences, and this method is highly sensitive, reproducible and reliable. Now if we talk about different molecular markers that are being used for analysis of somaclonal variations, and the commonly used DNA markers are RAPD, that is random amplified polymorphic DNA, RFLP, that is restriction fragment length polymorphism. Then we have certain microsatellites which include SSR, simple sequence repeats, ISSR, that is inter simple sequence repeats, then STR, short tandem repeats, then AFLP, that is amplified fragment length polymorphism. Then we have SCOT, that is start condom targeted polymorphism. So all these molecular or DNA molecular markers can be used to detect, to analyze whether the somaclonal variations have occurred or not. Now every molecular marker has its own advantages and limitations. Now discussing these molecular markers in detail will be beyond the scope of this lecture, but I will just give you an overall comparison of some of the advantages and some of the limitations of these markers. Now if we talk about RAPD, then the method involving usage of this marker is quick. It is a method of low cost, but the limitation is the method is having low reproducibility. Similarly, if we are using ISSR as a molecular marker, then we obtain better reproducibility and we consider it as a microsatellite based method. Again, SSR, it is a highly polymorphic marker, it is codominant and provides you highly reproducible results. If you are using AFLP as the molecular marker, then this will provide you high resolution. It has a wide genome base and it is extensive and but it requires more optimizing than RAPD molecular marker. Optimization in terms of the methodology, in terms of the concentrations used, in terms of the analysis of the results. If you are using RFLP as the molecular marker, then the method becomes labor intensive. It is largely a probe based method which requires usage of radioactive material, so one has to be very careful while using radioactive material, so this becomes a limitation of this marker. If you are using SCOT as the molecular marker, then this marker has specific targets, specific genes as the target in the regions, but the method is moderately reproducible. So these were some of the advantages and limitations of different molecular markers that can be used under this method. But by and large, if we look into the advantages of using molecular DNA markers as the method for detection of somaclonal variation, then these will provide you more accurate and quantifiable results in comparison to all other methods we have discussed. Now we move on to the last aspect of somaclonal variation, that is its applications. Now since we are inducing or we are generating variations in the plants, so we can introduce certain desired characters as variations in the regenerated plant, and these desired characters can be in terms of bigger fruit size, more interesting and beautiful flowers. Such kind of variation we can induce, as well as an improved quality with better yield of metabolites can also be produced. We can also introduce genetic manipulation in the medicinal plants during such kind of somaclonal variation. Thereby we can enhance the production of the secondary metabolites from those medicinal plants. We can also introduce certain useful variations in the plants, and we can develop new plant varieties which are resistant towards diseases or different kind of pathogens. New varieties which are resistant towards action of different herbicides. New varieties which are resistant towards different kinds of abiotic stresses like low temperatures, which we discussed, salinity of the soil, drought conditions are there, presence of heavy metals in the soil is another abiotic stress. So we can develop varieties which are resistant towards all such kind of abiotic stresses. We can also develop new varieties which are resistant towards insects or different kinds of pests. This will help in reducing the overall usage of these pesticides or the herbicides during the cultivation of the plants, making cultivation more economical and profitable. Now, apart from certain applications, there are limitations, certain limitations of somaclonal variations. If the main aim of tissue culture is micropropagation, then induction of somaclonal variation is not desirable, because in micropropagation we mainly focus on developing of the clones. So in such cases somaclonal variations are not desirable. Necessary screening is required to screen large number of progeny of regenerants, because there may be certain random variations which are not desirable, and if such random variations are occurring, it becomes necessary to screen large number of progeny of regenerants. The process requires optimization. So if you want to have reproducibility of these variations in the upcoming generations, one has to first optimize the entire process, because such kind of reproducibility will depend upon many factors. Conditions is one of the factors, genetic makeup of the explant material or the regenerated plant is another factor. Likewise, there are so many other factors that will govern and that requires optimization. Sometimes it's very difficult to select an individual which is having such improvements in the next generation. So friends, these were some of the limitations of somaclonal variations. Thank you.